All right. Let's actually start the event if it hasn't already been started. It has not, so we'll start it now. Fantastic. So people will fill in now once the announcement has gone through. Already been started. It has not, so we'll start it now. Welcome one, welcome all. All right. Okay. I think we should get started. Okay, so um yeah, where should we start, Michaela?
You tell me. We're trying to troubleshoot the screen sharing right now. We have some network issues apparently.
We can see your screen.
Oh, you can?
We can see it. I mean, like
Mine?
at least
Or I'm in
yeah,
US?
I can see the Nova slide deck.
Oh, amazing. Okay.
Perfect.
We're gonna fly blind.
Not all of us can though. All right, so some people cannot see this.
Interesting. Okay.
It's about 50% here in the chat. Classic
Classic
Discord.
Discord, yeah.
So I mean, the nice thing about Discord also though is that if you have slides, you can actually just take a screenshot and put them directly into the chat. That actually works pretty well. People can go back and take a look at them. So if there's someone on your side that can do that, like for instance, I can actually do that. And then it will
Can
come
I
out.
send you my slides just in case?
Yeah, you can do that.
Okay, perfect.
It's usually a networking issue.
Oh,
Yeah.
just a second.
Okay, I've downloaded the virus. Great, and it's right. All right. So we've tried you leaving and coming back, right? So the turning off and on again didn't solve our problem.
No, the magic solution did not work this time.
Okay.
I think now it is going. Try to change to the next one very quickly, Amanda. Yeah, it's working.
Can you see? Cool.
Yeah.
Can the rest of Discord see?
No. Now I can't.
Okay, this is
Oh.
like really stealth operation over here. Only some people can see.
Yeah. So one thing that you can do, and I think just as good, honestly, is to just take micro screenshots and just chuck them into the chat and then talk over those screenshots.
Okay.
It's basically an equivalent to what you're trying to do.
Okay, we'll do a hybrid version. Hi everyone. Should we get started?
Let's get started.
Okay, so for those who didn't hear, I'm Micaela. I'm here with Pedro and Amanda.
Hey.
Erdolf is also tuning in, and we're from Metanova Labs. We're going to talk about Nova, which is subnet 68, and it's focused on drug discovery. And essentially, you might have seen us retweet today. CZ came out, was like, two most promising sectors in crypto are AI and pharma, and we're doing all three. All three have shown immense value creation, and in fact, merging the three may be the key to solving for their individual limitations. I think together they can be game changers. We know that companies in these fields raise tons of money and they're valued in the billions, but even that misses the core of our mission, which is to accelerate medical innovation. The impact this technology can have on people is hard, if not impossible to measure, but something to keep in mind as we build a feedback loop between digital and real world value creation. drug development has fundamentally changed society. Started with the aspirin in 1899. Since then, life expectancy has gone up 42 years. And even though that isn't a causal relationship, we now have the capacity to treat a wide spectrum of diseases and health conditions. So it's not a wrapper, it's not about tokens. We're talking about things that really impact you and the people you know. Global pharma revenue is estimated at $1 trillion, but the true value goes way beyond the sales. And this all sounds amazing. We know it's worked somewhat well, but it's currently in a state of crisis. On average, one drug takes about $2.6 billion in 10 years, and it has only like a 10% success rate. This high risk, high reward paradigm has led a lot of people to just focus on patent busting and doing small incremental changes to the molecular structure of drug compounds. We started tinkering with AI, not we, because I wasn't born, but people started tinkering with AI to predict activity in the 60s, but it wasn't until the last decade that we really had the ability to process enough data for it to make a difference. AI holds huge amounts of potential to make it better, faster, cheaper, more novel. The earlier you can shortlist molecules of interest, the more efficient the rest of the development will be, with each additional step increasing costs exponentially. So what AI really allows us to do is to tap regions of the chemical universe that have never been synthesized before and search for novelty rather than just changing atoms of compounds that we already know work. And we really think that that's where the breakthroughs can come through. The reality has been a little disappointing. And I think a huge reason for that, the reason why AI hasn't really delivered on its promise, it's because it's being done in the wrong way and the incentives are misaligned. So most of the models that are being developed right now are being developed by research institutions and private universities in a vacuum. And to actually gain any kind of predictive power, you need to train these models on huge, diverse data sets, preferably from real world data and optimizing for multiple parameters that increase the likelihood of a drug being safe and effective, which is ultimately what makes it approved and then sold. The early stages of drug development are commonly referred to as the valley of death, where most drug candidates fail, not necessarily because of scientific limitations, but rather economical and technological challenges. We believe Bitensor can actually narrow the valley of death more than maybe any other crypto network. And our subnet is allowing us to access early stage funding through the token emissions. Thank you, DTAO. That allow us to push innovation in a way that academics and other companies can't. Like we can be a lot bolder. We can actually go for the moonshot. We can actually go for the breakthrough. We can decide. It also allows us to tap into a global talent pool that we all know is super cracked. It sometimes feels like we're at war, but actually we're on a mission together. And we're all being rewarded by the same mechanism. So it really reduces the burden of having to raise all of that money to get into this field. The merit-based, like hyper-competitive model also means that miners are constantly optimizing for the shortest path to a reward without the bias of a medicinal chemist. And it's in this adversarial environment that we're able to really stress test the models and identify the areas of low confidence that compromise its predictive power. Overall, how does this work? We are using a data set from Savvy that has like over 1 billion synthesizable molecules. Synthesizable being a key word here because you will see a lot of other platforms that are operating in the theoretical realm, and that's just way less actionable. Like we haven't actually figured out how to synthesize every known configuration of atoms in the world. And that's what we're giving our miners. We've already seen over 500,000 downloads of that data set in the last month. And they're using that data set to run predictions on this protein ligand binding model and identify the ones that have the highest affinity or lowest affinity to a combination of targets and anti-targets. Validators are then using what's considered a state-of-the-art prediction model to score those compounds and identify the winner for each challenge. Based on those submissions, we're going to be able to do a lot of different things that we'll talk about in greater detail. But fundamentally, it's chemical maps, it's libraries of the most promising molecules, it's fine-tuning and retraining of models, and most importantly, down the road, synthesizing them, validating them, and both using that data to make our subnet even stronger and more robust, and to push our own R&D and that of companies and universities that decide to partner with us.
So ever since our launch on March 1st, we have run approximately 780 challenges, which we were able to explore 4,000 proteins, and these generated a set of 17,000 unique molecule predictions of binding affinity from these molecules to these proteins. During this time, we have iterated on our incentive mechanism frequently based on the paths of least resistance that miners found to win the competitions. And in fact, miners are extremely efficient at finding these paths of least resistance. And this has led to a series of updates that have been performed in order to guide the incentive mechanism towards a drug discovery problem instead of a blockchain one. So at first, we had one single target, and this has evolved into a pair of target and anti-target. And later, we have changed our competition format to predict the binding affinity of one target against a set of anti-targets. We have implemented encrypted submissions to avoid front-running and copying of answers. We have expanded the protein data sets to over 2 million proteins to increase unpredictability of what the next target would be. We have implemented a minimum required number of heavy atoms. Heavy atoms are those that are not hydrogen, so anything else that's not hydrogen. We have tuned the weight of target to anti-target in the calculation of the final score. And in fact, we are continuously optimizing these parameters that we use to calculate scores. And this has allowed us to collect data on certain pitfalls of the model predictions that are not commonly discussed in the literature, even for state-of-the-art models, because the creators of these models are not incentivized to do so. But these pitfalls are extremely important for the application of these models in the real world because they can translate in areas of higher uncertainty.
So for the ones that can see our screen, the next two slides are shared as 1 and 2. Now it's 1. So basically, when Novelty started running, the challenges covered submissions for just one protein target at a time. And very quickly, we noted the power of the swarm of miners in really finding the shortest path to out was being requested by the incentive mechanism. Miners they very quickly found a region of low confidence in the psychic prediction, as the data is showing us here. In blue, we have a representation of the distribution of Savvy's database atom counts for the molecules. And in cyano, you can see the prediction scores for affinity for a random subset of 300K molecules for one specific target. We tested this to check for this distribution across multiple targets, and the pattern is virtually the same. You have a high variance region in terms of psychic predictions for molecule versus target affinity that correlates with lower atom counts, even though the density of molecules, of this kind of molecule in Savvy's database is quite small, representing a potential hole in psychic's scoring function. This was not reported in the original paper, but miners found it really fast. As an answer to that, we implemented a progressive minimal atom count increase and target versus anti-target system, amongst other approaches. For the challenges, really with the aim to correcting for this kind of scoring hole, but this worked only up to a point. Now going to the next slide, when we expanded the data analysis of the committed molecules under this new target anti-target system, plus increasing minimal number of heavy atoms, we noted that miners found yet another hole in psychic's scoring function. The data presented here now represents a different molecular property, the number of rotabochemical bonds in the molecules. Cyano is the same data set of molecules, but now for the predictions of target minus anti-target. And the purple represents this information for winner molecule, okay? So yellow points here, they represent the winning count in function of the rotamers of the molecules, considering the best answers for 112 challenges. We saw that again. The miners discovered quite quickly that molecules with higher atom count but very rigid, with no rotamers, could also win the challenges, but without necessarily being what we would be looking for in a drug discovery context. Again, these molecules are really underrepresented inside this dataset, and they show quite interesting properties. They usually have pretty low affinity for most targets. Since they are so underrepresented inside, iterating over all of them to get the ones that would have a substantial difference in terms of like target minus the anti-target scores. In this new system would be much easier than going through the whole data set. Again, another unknown potential relevant hole in the prediction model if you are considering for selectivity of the molecules.
So the Shannon upgrade, which is about to be released tomorrow, which is Friday, April 11th, is a result of this process of the incentive mechanism refinement and data analysis on the winning molecules that we have been performing. So this incorporates elements of our current competition model. So we'll continue to use the 1 target versus 4 anti-targets format. One of these proteins will be fixed and rotating on a weekly basis, and we'll use these to create chemical libraries for targets of interest. And the other 4 proteins will be randomly chosen from the 2 million protein dataset, and we'll be using those as proxies for off-target effects and toxicity. And for this update, miners will be required to submit sets of molecules instead of a single one, and the scoring system will include a penalty for molecule repetition. And this has the goal of incentivizing miners to explore the dataset beyond the same chemical scaffolds that have been previously explored, and will also include a diversity bonus, which will be calculated with the Shannon entropy of the molecular representations of each miner's submission. The focus on chemical diversity for this update is aimed at leveraging the community's efficiency at finding the model's pitfalls in order to map the chemical space and to find regions of high variance in the predictions that will very likely be sources of uncertainty when you try to apply this model in the real world.
So a lot of people hypothesized for a long time that computationally mapping vast chemical universes in terms of activity profiles was indeed the path to improve the success rate in drug discovery and drug development, but the real data confirming it started to being released only this year. Now that in silico methods are really getting to the billion-sized level, and we can explore those huge chemical spaces, we're getting to know the power of it, as substantially improved success rates when going from simulation to chemical synthesis and biological validation. But also, we are learning the considerations that one must do when dealing with ultra-large screening predictions. For example, chemical regions that can game scoring functions even of the best models. Here, they are exemplified, for example, by lower atom count or rotable bonds, features that miners can detect with unprecedented efficiency.
So we're gonna map the chemical universe by regions, and our first map is called TREAT, Targeted Reward Evaluation and Therapeutics, which is gonna focus on target systems associated with reward and learning mechanisms that are key for people to be able to regulate behaviors such as focus, eating, connection, rest, sleep, overcome trauma, list goes on. That's because we are particularly interested in what we're calling meta-programming drugs, meaning we want to train the AI to develop drugs that allow people to train themselves. And actually, there's a lot of knowledge that you gain in both exercises that can help one another. And there's a scientific reason for this. Like some of the longevity interventions that have the strongest backing involve lifestyle modification, like caloric restriction or sleep hygiene. Behavioral regulation is fundamental to health and well-being, and it's probably more effective than just waiting for you to like get a disease that you then have to treat. Metaprogramming might sound a bit out there, but if you translate it to the traditional disease-centric framework that's used by regulatory agencies, you get therapeutic indications like ADHD and sexual dysfunction, and blockbusters like Adderall and Viagra. And we think we can do better than give meth to children and solve for intimacy issues by increasing blood flow. Nova is really like planting the seed for medical innovation that goes way beyond than the subnet, way beyond than us. Like we really think that we can be the starting point for a giant flywheel of digital and real world value generation and build feedback loops between it that make each more robust. Fundamentally, all of these will be works in progress, and our idea is to do those to support our own R&D, but also have been actively in conversation with different partners. In fact, have already been approached by one that is interested in us doing this for them. In terms of next steps, we're going to be launching officially the Shannon upgrade tomorrow. Thank you to everybody in the community that gave us their feedback on the code that we posted earlier this week. It was super helpful. And alongside it, we're going to be launching the treat challenge, which is going to be guiding the curation of the fixed rotating target. We're also working on releasing new prediction models that are based on benchmark and retrospective data. And over time, we will be integrating more properties, ADME talks, other properties that are key in developing a drug that actually works and that can get approved, such as crossing the blood-brain barrier or like certain levels of lipophilicity, etc. And all of this will lead us to creating libraries of really high value, especially once we're able to like validate some of the top compounds in the real world through synthesis and wet lab assays. And we're advancing 3 different partnerships. We think those will be really key in building this bridge between crypto and pharma. And it will require us to have a hybrid approach as well. I'm not going to tell you that in supporting Nova, you will save a life, but I can tell you that there's a non-zero chance that it will. And in this way, we're very excited to be what could be the first crypto native biotech company. And we really believe that DTAO is giving us the opportunity to build that bridge in a way that others haven't been able to. And that's all we got for the
Awesome.
presentation.
Thank you. Let's zoom in because there's so much here. You really have thread the needle all the way from the tech and funding and time lock encryption and stuff, all the way to hey, let's save lives. But let's like zoom in into each one of these sections. As usual, where I'm most interested in the actual verification system, the validation system behind Nova. Which, can you dive into this, Amanda, and explain like why does this validation system work, and what are the pieces and yeah.
All right, so for our update, we noticed that in previous challenges there was high levels of occurrence of the same chemical scaffolds. So we wanted
No,
to
let's back up.
include.
Let's back up a second and all the
Sure.
way to the beginning. So like, how does Nova work on BitTensor?
All right, so the challenges, they work by providing one target and now 4 anti-targets to our miners, and they need to find molecules that maximize the difference in binding prediction scores through the target score minus the average of the anti-targets. So they
And so
aim to maximize difference.
And so affinity is like if I give you a molecule and a protein, we want to know whether or not those things combine, if they're going to interact with each other, correct?
Exactly. And we do that with a model that's considered state of the art nowadays. It's called Psychic. And it works by predicting with the SMILES chemical representation of the molecule and the amino acid sequence of the protein, whether or not the interaction between these two entities is energetically favorable or unfavorable.
And this is important because when you're trying to discover a drug, you say, "Hey, this is the molecule in the drug. I'd like it to hit the this protein," or is it the other way around?
No, usually you have
Protein.
huge possibilities of what a molecule can be. So you have billions of possibilities, trillions. Like
Yeah,
it's
I think on that, you usually want to find molecules that are going to be selective, so binding and having the effect that you want them to have over specific protein targets, but not doing that with what we would call off targets that could be involved with side effects. So
Right. So I want to find a molecule that if I take this molecule and it crosses the blood-brain barrier into my mind, it will activate GABA receptors, but it's not going to activate this other thing that I don't want. And those would be proteins. So I want to find affinity I want to find a molecule that has high affinity with this protein that is the thing I want for searching the neurological space, changing my mind, improving my day, making me happier. But I don't want it to, affect my other aspects of my personality.
Correct.
exactly.
or for biology as a whole. So the selectivity feature is really, really important. Indeed, a lot of drugs under development in clinical trials, they fail because of that. They might have chemical features that make them like prone to bind to unknown proteins. And this is linked to like side effects that you usually do not want, or they might be linked to the drug. It would bind well to the protein, but it don't get to the region in the body that needs to get. So there are a lot of properties that we want to optimize, but a huge part of that is optimizing for the binding to the right proteins and not binding to the proteins that you don't want them to be binding to the off targets.
Yeah, in
And
order
so,
for a molecule to act upon a protein, it needs to interact with it, it needs to bind to it.
and how big is this problem? So, in the process of doing drug discovery, is this the largest computational problem that you need to solve? You need to know as quickly as possible and iterate as quickly as possible across as many molecules as you can think of to find the right proteins that they'll bind to and the ones that they won't bind to in order to do drug discovery. So if this machine really works incredibly fast, faster than anything else in the world, it means you're actually speeding up the processes which we can search the space of drugs, correct?
Exactly. And not only that, but if it is scalable, highly scalable, you're also finding a lot of chemical entry points to do what you want to do. If you have more chemical diversity to activate the target that you want to activate without interacting with off targets that are linked to side effects, you have much more possibilities in terms of really getting a drug that is gonna do what you want it to do.
Yeah, and you can
And so,
filter for side effects a lot earlier in the process as well.
right before you actually develop the drug and put it out into trials and use rats or whatever mice to test the actual drug and its effects, if you can do this way
Right.
before, you can iterate faster and we can get to the quote unquote limitless pill, which exists out there in computational space. We just haven't found it yet.
Yeah, the process is often referred to as a funnel, and the narrower the top of the funnel is, the more efficient everything else will be. And so that's why we're starting here and then building on top of it the other properties, because if we can optimize for all of them together, we are going to be able to have a way higher success rate. I mean, it's a bold claim, but it's like fundamentally true.
Yeah, yeah.
And
This is folks.
how much are drug companies expending on this problem? And like what is this problem worth to them?
Hundreds of millions of dollars.
Yeah, this is becoming actually the next frontier to really change drug development. Because if you can do that prior to synthesizing and doing biological assays, this means a lot of savings in terms of the whole process.
Right. So if a drug costs $2.6 billion, in large part is because a lot of the candidates failed along the way. So you were very inefficient in how you were using those resources. The problem that we're seeing is that you have companies raising hundreds of millions of dollars to do this because their valuations depend on the privacy of those models and the fact that other people don't have access to that data. When you integrate it with Tensor, then there's actually an incentive for there to be more data sharing, which allows you to explore larger parts of the molecule data set and in general, like identify more entry points. So we're focused on our own R&D, but we recognize that this is helpful for anybody that's it's in the industry at large.
Because they can come, if you're a researcher and you're trying to develop a new drug to cure this type of cancer, you have to go and do this computational process at some point. And what does it look like in the field? Like, okay, I want to test this thing. You send it off to the cluster at your university or inside Pfizer or whatever, and you come back in a day and a half and check the results, and it's maybe not that great.
Actually, these kind of screenings take usually more time. You can see like screenings going from like at least weeks or even months if we're talking about billion-sized data sets.
Right. So if you can speed that up by tenfold, it means that we can potentially get the drugs 10 times faster. It's just a matter of iterations.
Yeah, and the miners are already operating at lightning speed. And that's why, like, at first we were trying to direct a problem, and then we realized, like, this is a feature, this isn't a bug. And in fact, this is a question that nobody else is allocating resources to. But in having this different relationship and different roles to play within the subnet, they're immediately identifying all of the weaknesses in these models that then allow us to have more robust predictions for the ones that we do want to have reliable predictions on.
So let's talk about this. So you guys, you basically challenge the network with a problem that would look like the problem a research scientist at Pfizer would want solved. Hey, I'm trying to hit these proteins, and I don't want to hit those proteins. Let's try this molecule. We just, we thought this up in our minds. Let's try this molecule. Well, let's try another molecule. This is the thing that they're trying to do. And so every challenge period on the network, you basically construct a synthetic version of that problem and have the miners solve it. And is that correct?
Yeah, that is correct.
And so what are you guys seeing now? Like, has the network been improving? Like, are the miners blowing you away? Are they ruining your lives? I believe
This
that was
is
a slide.
actually
Yeah, go on.
this is actually being something really, really, really interesting because miners are finding holes in the scoring function of this state-of-the-art prediction model absolutely fast. This is actually a huge feature. The fact that we can detect these emerging problems that come from scaling to billion-sized dataset predictions, this is a huge asset that I honestly have not seen anywhere else. Usually, you're going to be looking for filtering for this kind of scoring holes later. And the fact that they can do that so efficiently at the very beginning is really, really good.
But yes, we've been releasing lots of updates in order to steer the problem into something that becomes more of a drug discovery problem instead of, who submits the molecule faster or, things like that are very blockchain types of problems. But really, it's like Mikhail said, it's a feature, not a bug, because knowing where your model doesn't work guides you away from these regions of high uncertainty, and it's super valuable for developing a drug asset in the real world. And the creators of even the most advanced state-of-the-art models are not incentivized to do this type of stress testing in their models. They want, beautiful data for the publications and stuff like that. So having the community stress test these models is a huge feature, and it's super valuable.
Is there like a flywheel here that you guys can imagine with respect to the answers produced by the miners, finding holes in that model that you're using for verification? I think you said it was called psychic, and then training or producing a better model that can go into the verification side of this. Like, how do you connect that loop together so that this process, has a flywheel effect that can continue forever?
Yes, this is in our next steps planning of fine-tuning psychic versions for specific targets of interest that we want to develop for. And if you take a look at this slide, for example, you can see that the regions of highest variance are usually in the least represented data in this case, it's a prediction data set, but you can use it as a proxy for the whole chemical space. And when you have very low represented regions of the chemical space, your model's predictions are going to be less accurate. And yes, this is something that we want to do, like in the near future, is fine-tuning psychic versions that have a modeled training set in order for us to get predictions with less variance in the areas that really matter, in the areas that are drug-like.
So these charts here are showing potentially the creation of a data set that can be used to improve psychic.
Correct.
And how do you filter this data, or like what does that training algorithm look like? Like fit this better, or like what exactly is the learning process that would teach psychic to be better here?
So there are two paths here that we're going to be taking in parallel. One involves fine-tuning the psychic with known data that was not included previously for the original training of psychic, in terms of molecules and their activities for targets of interest. And the second involves us generating lab results and also feeding it to the model. And doing this in parallel, we believe is the key to really unlock the access to the key maps in the chemical space that are going to be elucidating the pharmacology for targets that are highly relevant.
Right now, you just have a single model, but I remember speaking to you before about how the structure for this subnet um, is sufficiently composable that you could just put any model in that, or an additional model, or multiple models that predict, not just affinity, but any type of structure of the molecule, or uh, you know um, other qualities. Like, can you talk a bit about that? Like, where do you guys take what you have is a very like constrained problem, very simple problem, very important problem, but how do you go to the class of larger pharmacological problems?
So one path would be fine-tuning psychic for specific target family. So for example, enzymes or membrane receptors or ion channels. Another path would be modeling other pharmacological properties. So we've been developing a blood-brain barrier permeability model. We're really excited to share this with the community soon. And this is one of the predictions we would like to incorporate into our predictions, because this is something that's very relevant to the TREAT initiative, because we're focused mainly on psychiatric drugs. So they need to cross the blood-brain barrier, and it's a super complex property to model. But other possible properties we can model are pharmacokinetic and pharmacodynamic properties that are relevant and that sometimes may cause, and frequently cause, drug candidates to fail clinical trials because they suddenly show unexpected effects on, for example, metabolism or other important functions that they're not supposed to interfere with.
In
Right.
very simple terms, your drug needs to get to the right tissue in the body. And ideally, it won't be binding or interacting with off targets. So indeed, this needs to be a sequence of models.
Right. And the reason why we chose psychic is because it's very generalizable. So it's like a great starting point. But the subnet is being designed in a modular fashion where we could even swap psychic for a different activity prediction model, and add more for the miners to optimize against. And really what we've noticed with the miner behavior is that this can be a way for us to optimize any open source model that's been underfunded and turbocharge what's already there. And it's not really being used because it hasn't had the funding to be reliable enough, but well, here come the miners, expose the weaknesses, and we can create a new wave of more robust open source models that were trained on Tensor.
What do you think the miners are solving here? Do you think that what they're doing right now is just scaling compute to run more iterations and do more checks? Or do you imagine or even know what type of optimizations, intelligent optimizations they're applying in order to do this better and faster than any other competitor in the network?
So we discussed a lot about that, and very likely we have a mix of strategies here. Certainly, they are doing like a relevant amount of predictions, pretty relevant, to get data, and then with this data, be able to find what would be shortest paths to winning the challenge. I think honestly, they are mixing large-scale predictions and deep exploratory data analysis to find key patterns that in a very good way also align with potential holes in the scoring function of the model that we are using, and uncovering those at the very early stages of this scalability is essential.
I think the main point of contention was how much of the data set they were actually leveraging. And that was the big motivation in introducing the Shannon upgrade, because diversity is incredibly important in us having high value outputs. And so incentivizing them with diversity bonus scores will make them look farther and wider than smaller subsets that will just become like repetitive and not necessarily push us in the search of novelty.
At the same time, getting to know what are those very reduced subsets requires them to really go over the data set multiple times.
So I know you guys are not just here to do incentive mechanisms. Like what brought you to BitTensor was the power of the machine that would allow you to achieve your goals, really, of helping humanity generate these drugs. Where do you see Nova, the subnet, going? And this is one of the questions in the chat. Next, like it works for you. So let's say that it works. You're doing this amazing research. Like, what's that next stepping stone for you as a team?
I mean, I think there's a lot of parallel tracks, and the subnet is like the portal to all of them. And there's a parallel track, or there's a line that we follow that's just more traditional, like drug R&D. And then there's the portal that can be used by everyone else. So we're not the only end user of this subnet. Does that answer your question?
Of course. And what would you want to use it for, first of all?
Well, first we're going to be improving the models, because the more we can optimize the advanced virtual screening process, the better we will be able to like take on our own R&D ambitions, and the more attractive it will be to other people that are interested in this kind of problem.
What does that look like?
Well, it looks like starting with the retraining and fine tuning of the models, and then the thing that I think will really allow us to reach escape velocity is once we are able to benchmark those with retrospective data that the model has never seen, that essentially is like a proxy for its accuracy. And then from there, synthesizing it, like actually jumping out of the screen, entering the real world, having these molecules being tested, and therefore validating all of the work that's being done to date, improving the work that we will do thereafter. And I think have, reproducible data that we can share with more traditional partners that are potentially skeptical of crypto, but that will trust results from the same laboratories that they are used to using.
Okay. So out in the real world, user feedback. Basically, you're going to make drugs and take them yourselves. Um, so this machine that you've built, where do you see the monetization coming from? Like you guys can build, a first class pharmacological company, but
Hmm.
others can also use the network, right? That's the power of Bitins or subnets is that they're open and they're actually completely open source and open access. Um, are you said you talked a little bit on your last slide about partnerships and other individuals and teams that you're working with. Can you talk more about that? Like, how are you getting this early use?
Right. So, I mean, we're leveraging Pedro's experience in the field and the network that he's built over time as well. I think that puts us in a pretty unique position in this side of the world. And really, it's about monetizing everything that we think is moral to monetize, but also like waiting for the right amount of validation for it to be really worth something. So first step is definitely partnership. It's more of like a service model, but once we have more and more data that verifies the predictive capacity of these results, like we will be able to like monetize things that are at a smaller scale, like for example having token gated access to these curated chemical libraries.
From the chat.
It's about working on, there's different development timelines. Like every single day, we're pumping out potential candidates in theory. But the farther you take them down the development path, the more they are worth, exponentially so. And so it's just a matter of making sure that you're capitalizing on what has the most verifiable value at that given point in time.
From the chat, a couple questions. From Brian, how do you collaborate with industry and have them be okay with having their data and ideas open sourced? Is that actually a legitimate question or a requirement of the design you guys have?
So one key point on that is that there is also a relevant amount of analysis that needs to be done after the output are generated in terms of the maps, right, for activity for the targets of interest. So we can also be building on top of what is being generated in the open source realm, focusing on bring this level of like enough differentiation for the interaction with more traditional pharmaceutical players.
So this is to say that you're not worried about the data being open that others use. The
No.
network. But
Yeah,
even if it were open source currently, it would be possible to do encryption and encrypt the answers for a select number of keys that would be hard to access for those other competitors in the space.
Right. And the more validation these results get, the more they are worth, right? So even if you think about it as like free and premium option, like there is value in being able to showcase to people the work that we are building together. And then there's maybe an extra layer of effort that can be considered like the premium. But we want to be able to appeal to both and really bring these now disparate industries together because it really seems like they have the answer to each other's problems. Like crypto is being criticized for being scammy not having real world value, pharma is, in a state of crisis, as I mentioned. Is there any way that they can come together and do something? And it's gonna take people from crypto to build the bridge.
Crypto is the solution to everything. Question from the chat, kind of a funny question. Where's the sign-up sheet for the Metanova human trials?
We'll keep you posted.
Just put it in the
We'll
chat here.
definitely add a form.
The interesting point that, if we can make this more open and people can actually take a bite out of the ability for drugs to be generated, do you think that solves some of the problems in the pharma industry? And what are those problems that you see right now?
Time, cost, lack of novelty, no incentive to go after something that hasn't been heavily validated. If you talk to a biotech VC, they're like, bring me clinical trial data. And there's no real explanation as how you even get there. And it costs a lot of money. And so it means that we're like moving like a herd from one disease to the next, depending on how much the drug developed for it has sold for. It doesn't necessarily mean like we're matching our needs with the kind of capital that we're allocating for development.
From the chat as well. There's a question about synthetic data. I'm going to jump to Fish's question. Can you please elaborate on your 3 partnerships that you mentioned? I didn't catch who the partnerships were with and why.
I can characterize them, but they are all sensitive about being called out by name, especially prior to an official like announcement. But one is a research university, the other is a private company, and the third is a CRO, which is a contract research organization, a lab.
Okay. Hopefully that
It is
answers
quite standard
for Fish.
in this kind of interaction for these negotiations and discussions being under confidentiality until we all talk about them openly on a press release.
Okay. I'll try to, I won't press on that then. Guys, we're at the end of the call here. It's been fantastic speaking to you. Great presentation. Unless there are any more like major questions from the chat, we can end it here. Or if you guys have questions for me, or if you want to bring stuff up on stage, let me know. I agree. One of the best researchers. Great presentation, guys, and awesome work.
Thanks,
Like
Guy.
this team is only a month in, and they have a functioning exploit-free incentive mechanism on BitTensor, which is absolutely incredible. They're using T-lock, as I understand, which is amazing. And once you get to that point where you're not just being attacked by minor issues and you're actually focusing on real science, that's really a watershed moment for a subnet. And congratulations to getting to this point and being up on Novelty Search. Fantastic work to the 3 of you.
Thank you, guys.
Great, guys.
Thank
All
you.
right, let's end it here.
Thanks, guys. It's been-
Ciao, everyone. Have a good night.
Ciao,
Ciao.
ciao.
Yeah.